Good evening, good evening. How is everyone on this fine Monday evening? Great, that's good to hear, Max. Excellent, Julia. Hi, Asma. Great. All right. Well, it looks like we're split about 50-50 between taking this in May or taking this in June or later. So welcome. If this is your first channel session, welcome to the channel. If it's not, welcome back to the channel. So today we're going to be working on the cell. This comes from chapter 1 of your biology book. So please make sure that you are comfortable with that, because what we're going to do is go into extra information. So let's get into this. We have some very specific learning goals for today. And these learning goals are things that are vital and very often tested on the MCAT. So we really want to make sure that we have this science knowledge under our belt. We're going to recognize and identify the different functions of eukaryotic membrane-bound organelles. Okay, we're also going to be looking at cytoskeletal structures. We're going to learn to classify prokaryotes and identify the structure and functions of a prokaryotic cell. We're also going to start looking at the growth and reproduction of bacteria. Some of this is going to include how they transfer genes between each other. All right, so our first learning goal, it is imperative that we understand the membrane-bound organelles in eukaryotic cells. This is a high-yield topic. The MCAT absolutely loves this. So we're going to jump into this and really understand what those different membrane-bound organelles are, as well as the cytoskeletal structures that we need for, just the everyday cell life of being able to do things. So first of all, we're going to start off with our nuclei. So the nuclei, again, has a membrane, so it's a phospholipid bilayer, just like our outer membrane. And what it does is it separates out and it contains all of that genetic material that we have for cellular respiration. So what we're going to do is we're going to zoom in on this part right here, which is really the central part of that cell, that nucleus. And we're going to talk about a couple of different things that are important. The first one is the nuclear pore. So the whole function behind the nuclear pore is to regulate trafficking of what comes into and out of our nucleus. Okay, so we need to really make sure that we're picky about what goes across back and forth from that cytoplasm. And that's the function of our nuclear pores. Our next one that we're going to talk about is going to be the nucleolus. You guys tell Eli and I, what is the function of the nucleolus? We can see that when we're looking at cells under a microscope. It's a darker spot within the nucleus. Yeah, it doesn't consist of DNA. Has to do something with ribosomes. So Ali, yeah, it has something to do with ribosomes. So this is where specifically ribosomal RNA. Okay, so remember, ribosomes consist of both ribosomal RNA as well as proteins. So the nucleolus, this nice dark spot that we see right here, that is where our ribosomal RNA is transcribed. Yeah, is actually made. Good morning, Kay. All right, so this is where our ribosomal RNA is synthesized inside that nucleus. Then we have transcription. And transcription is inside the nucleus, and this is taking our DNA, making it into message RNA so that it can go and it can be translated into a protein later. So we go very much into that when you look at the molecular genetics channel sessions. We really talk a lot about how that works and how that DNA to RNA to protein synthesis works. There are some other pieces in here that we need to look at as well. But we're going to talk about those as we go through the rest of the slides. So the next thing that we need to talk about within the nuclei is really what that DNA looks like. So we've got our chromosomes, and these, when we think of a classic chromosome structure, this is the chromosome in its condensed form. That's the form that we see right before we go through mitosis, right before we go through meiosis. So this is that nice condensed form. So when we have our DNA our DNA is wrapped around these nice cores right here. What are these blue circles? What are these blue circles that I have my DNA wrapped around to really help me compact it? Yeah, exactly. Those are the histone cores. Perfect. So in a eukaryote, we have to organize this around those histone cores. Now, within the histone core, we have this euchromatin and this heterochromatin. When we look at euchromatin versus heterochromatin, what does it mean to be active? Why do I have spaces in between those different histones? This is where transcription is happening. Very, very well done. Because let's think about this. Though all of our cells have all the same DNA, not all the cells are going to transcribe the same DNA into mRNA. They're not going to all be using the same genes. So this is what we mean by active, is this is where we're actually doing that transcription. Then our silent over here, well, that means that we're really not doing anything with that heterochromatin. It's very nice. It's very compact. It really doesn't have anything that needs to be transcribed either at all in this cell, or it doesn't need to be transcribed at the moment. All right, great job with that. And then finally, when we look at this again in mitosis, we get these nice condensed chromosomes. These are the things that we think about when we're thinking about sister chromatids separating during mitosis. All right, so that's all we're going to do with the nucleus. Now we're going to switch gears and we're going to start talking about the mitochondria. We all know the mitochondria, this is the powerhouse of our cell. This is where all of our ATP, or the vast majority of it, is made. This is where we get that energy from our cell. When we talk about the mitochondria, we always talk about the fact that it has a two-membrane structure. So cells have mitochondria all around inside the cytoplasm. What we mean by the two membrane structures, we have this nice, even outer membrane. And then we have this nice, curvy inside membrane. What is this inside membrane, that inner membrane, used for? What's that inner membrane used for? It contains the matrix, so the matrix is in the middle of that. And the membrane itself is where our electron transport chain actually occurs. Very, very well done. So this is that double membrane that we're always talking about when we go through this. When we have this, it has oxidoreductase. And what this does is this catalyzes those redox reactions that happen to occur in the electron transport chain, which you guys just mentioned. So that's what's occurring across this inner mitochondrial membrane. We're not going to go into detail about that, but there are metabolism classes that go into very, very specific detail. This happens to be in metabolism 2. As we look at this, we know that cytochrome C, if I say cytochrome C, you guys should be immediately thinking about looking at the electron transport chain. But cytochrome C and calcium, okay, so it's not just cytochrome C, but it's cytochrome C from the electron transport chain and calcium, can be released to cause apoptosis. So what does apoptosis mean? If I say we're going to cause apoptosis. Yeah, that's that part of that programmed cell death within a cell. Perfect. You guys got it. All right, next, what we're going to be talking about is the fact that they have their own genome. What does their genome look like? The mitochondria has its own genome. What does it look like? Definitely comes from mom. It definitely comes from mom. Absolutely. And it's circular. It's circular, so it looks just like a bacterial genome. And this leads us to the set theory, this symbiotic serial endosymbiosis theory. Okay, so serial endosymbiosis theory is the set theory. And what that says is the inner matrix, that inner membrane in the matrix, was actually an aerobic bacterium that slammed into a eukaryotic cell. So the outer membrane is from that eukaryotic cell. And that's why it's got its own genome. And that's why it looks just like a bacterial genome. It's that circular prokaryotic genome within that cell. Now, these also replicate just like bacteria. How do bacteria replicate? And we'll talk about this a little bit more later, but what is the term for how they replicate? How do bacteria or prokaryotes replicate? Yeah, binary fission. Exactly, exactly. Straight by binary fission. All right. Now, Tom mentioned maternal DNA. So that puts us to a point that mitochondrial DNA is really only given to us through the eggs. It's maternal. We can trace this mitochondrial DNA back through mothers. Why is it that it only contains, it's only from the maternal? Because we know dad have mitochondria too. Dad had to have had mitochondria. So why is it that it's only maternal? There are actually two different things that can occur here. Yeah, Tom, perfect. One of the things that occurs is if that mitochondrial DNA does get into the egg cell, then mother cytoplasm is absolutely going to kill that paternal mitochondrial DNA. And so they do have some mitochondria, Lauren, in them, but it's really in the tail. So a lot of it gets left outside of the egg cell, so it never actually gets in. But if it does get in, we've pointed out that, hey, that egg cell is going to kill that maternal mitochondrial genome. Okay? We know it's killed because we can only have one form of mitochondria, Monique, within that cell, and that's always going to be the maternal mitochondria. Okay? So it's kind of it's almost in a way of protecting itself. All right? Good. Great job with our mitochondria. Now let's talk about the ER. The ER are membranes that are continuous with the nuclear envelope. So things can leave the nucleus, go straight into the ER. When we talk about the ER, there are two types of endoplasmic reticulum. What are the two different types of my endoplasmic reticulum? Smooth ER and rough ER. Absolutely perfect. So smooth ER, when we're looking at that, does not have ribosomes. Excuse me. And if it doesn't have ribosomes, what is the function? Thank you, guys. What is the function of my smooth ER? Some of it's detox, absolutely. Some of it's detox, and the other is lipid synthesis. Good. So my rough ER has these ribosomes on it. That's why we call it rough. We see these ribosomes all over it, which means this is where we absolutely are going to be making proteins. These are specifically secreted proteins that we are making as we go through. Now, well, these secreted proteins and these lipids that we're making, what happens to them? Well, they're going to be delivered via vesicles to our Golgi apparatus. What is another name for our Golgi apparatus? FedEx, absolutely. FedEx or the the UPS, the processing shipping center of our cell. Okay, good. So let's talk a little bit about this shipping center. So this Golgi apparatus is going to receive lipids or proteins that are made within the ER via those vesicles. When it gets it, it's going to modify, because not all proteins are made exactly the way they need to be. Some of them need to be modified, they need to be methylated or demethylated or have other side groups added to them. So they do that protein modification. Then they secrete those proteins in packaged vesicles. So when we look at this, we see a cis face and a trans face, which we're going to talk about in just a minute. Okay, notice on the cis face you have those incoming vesicles, and those incoming vesicles are going to be coming from our smooth and rough ER. So as we look at this, we're going to start out with our cisternae. Our cisternae are those foldings, okay, and they increase the surface area. We actually use the same term of cisternae when we're looking at those invaginations within the mitochondria. Okay, so it's the same idea. It's really increasing surface area. You'll also see cisternae used to talk about the invaginations in the brain. Okay, so the whole idea here is by having this, we're increasing surface area. That's the whole function behind these cisternae. Then next we have our lumen. Well, lumen, we all know what that means. That's that space inside. Okay, it's the same as saying the lumen of our gut or the lumen of our nephron. Okay, so that's that opening. That's where things are going through. This is where our modifications are actually going to occur. Okay? And these modifications, again, can be methylating, demethylating, adding carbohydrates, adding phosphates, adding sulfates, doing whatever we need to do to make sure that if it's a protein, that that protein activates the way that it needs to activate. Okay? Then we get our cis phase, and we already said we've got these incoming transportation vesicles. So these transport vesicles are bringing those lipids or those proteins that were just created in the ER, bringing those into the Golgi so that in its lumen it can do all those modifications that it needs. Then finally we hit our trans phase. Well, after it's gone through all of the lumen, after it's created everything that needs to create, modified everything it needs to modify, this is the side where the Golgi apparatus says, okay, I modified, I just packaged it into a nice vesicle, and this is where the FedEx part comes in. Now that I've got you prettily packaged, I'm going to send you off to wherever it is that you need to go. So cis phase, we all know cis means that it's on the same side, so that's going to be the cis phase coming from the ER, and the trans phase is going to be leaving the cell. All right. With that said, let's go on to another one. If I say lysosome, what is the first thing that pops into your head when Eli and I mention lysosomes? Christine, kill, kill, kill. Die, absolutely, Lauren. They're killers. They degrade, they break down, they destroy things. I love it. You guys have the exact right idea. Yes, it's the exclamation point that absolutely made that. So the lysosome enables the cell itself to recycle biomolecules that are within that cell into new cellular products or to kill things. So when we look at this, we have our lysosome. Here's a great picture of the lysosome right here. Nice thing about the lysosome is to kill or to degrade or destroy, however you want to put it, they have to have some sort of hydrolytic enzymes inside them. Well, gosh, we have to protect the rest of the cell from that because we don't want to be hydrolyzing things within the cell that we actually need to be using. So we do this via that bilayer. Okay, so it's got that membrane around it. Okay, and what this does is this helps us to maintain an acidic pH. It also helps to make sure that we're really separating, we're really protecting that cell. Okay, because we don't want to have that damage. Now, if we have one lysosome that happens to lyse or pop within the cell, because the cell is at a neutral pH, it really isn't going to be a problem. But lysosomes can be used for that programmed cell death as well. And what that means is we're going to be lysing a lot of lysosomes, and we're going to be lysing them all at once. At that point, we can have them work with apoptosis. Okay? Now, sometimes lysosomes don't actually work. So we're going to take this into a clinical setting here. And we're going to actually talk about Tay-Sachs disease. Does anyone know what Tay-Sachs is? You may have heard the name and you may not know it, and that's okay. But if you have heard of it, it is definitely a genetic disease. Agreed, Tom. Okay? It's definitely a genetic disease. And what happens is if there is something that cannot be broken down or the enzyme is bad, okay, it causes a problem. Yes, I agree, Navina. It is something that is more common in the Jewish community. Okay? So when we look at Tay-Sachs, what happens is there's a lipase enzyme that is defective. Okay? And we all know lipases break down lipids. Okay. So what happens is it has a defective lipase and certain cell membrane components start to aggregate in the brain. Okay? So this is where we actually have lysosomes that have lost their ability to be lytic. Okay? So it's a great job that you guys know that it's genetic, where it comes from, where it's most common, but definitely know that this is a problem with lysosomes, and it aggregates cellular components in the brain. Let's talk about another membrane-bound organelle. This one's easy, peroxisome. Peroxisome, by its name, means that it's got to contain hydrogen peroxide or other peroxide derivatives. Well, we know that peroxides can easily break things down. Okay, so again, we need to protect our cells from this. And this is why we have that lipid bilayer, that membrane that's around it, and then we have this nice crystalline core in the middle. The whole idea behind this is to break down long fatty acid chains. And we can do this with beta oxidation, and we can use it to build other molecules, or we can use it to make acetyl-CoA. This is something that is gone into detail about what beta oxidation is and how we use that to make other molecules or how we can break things down for acetyl-CoA. We see this in the third metabolism session from the channel. Okay, so that's another great place to go to get more information. So what we do is we have these nice fatty acid chains. So, well, what's going to happen to these fatty acid chains? They're going to come inside our peroxisome. And our peroxide, in this case, we have hydrogen peroxide specifically being shown here, is going to come along and is going to break this up. It's going to do beta oxidation. We know hydrogen peroxide can oxidize. Okay, so this is going to do that beta oxidation. This is found in eukaryotic cells. And one of the things that we can do with this is we can make acetyl-CoA. Okay, so we break down these fatty acid chains and we create acetyl-CoA, specifically from things like triacylglycerols, which is a very, very compact form of energy. Now, if this acetyl-CoA comes and leaves this cell, where in heaven's name is it going to go? Or leaves not the cell, but leaves the peroxisome. It's going to go to another part of, and specifically a membrane-bound organelle within that cell. Goes to the mitochondria, absolutely, because acetyl-CoA is essential for which part of my cellular metabolism? Yeah, TCA cycle, Krebs cycle, citric acid cycle, however you want to put it. Okay, so it's really important to know where that acetyl-CoA goes. This is the whole idea when you're starting to see this when you do your metabolism sessions. This is our beta oxidation. This is how we make that acetyl-CoA. And this can definitely go into our aerobic respiration, go into the matrix of the mitochondria. All right, so this pretty much covers our membrane-bound organelles. Now we're going to turn around and we're going to say, well, gosh, what gives our cells structure? What allows them to be the shapes that they are or allows them to interact with other cells? Okay. Now we're going to start talking about microfilaments and microtubules. Okay, so microfilaments are polymerized actin fibers. Wait, if I say actin, what's the first thing that pops into your head when I say actin? Oh, muscle cells. Yes, and it's specifically the thin part or the I band in our sarcomeres of our muscle cells. Absolutely perfect. Just for fun, what is the thick filament within our muscle cells in that sarcomere? Nice, myosin. So what happens is microfilaments are used to be able to move things around is one of the things it does. So our actin moves across the top of the myosin during that power stroke. Perfect. Now, let's also talk about what they do. One of their functions, if we look at this cell, it's a very, very specific shape. And microfilaments are going to help us to resist compression within that cell so it doesn't get squished. And it also resists fracturing of the cell. Okay, if our cell is fractionated, then it's going to lose its integrity. The cytoplasm and what's inside that cell are going to come out. So one of the things that it does is it regulates that shape. It gives it structure and helps it to not be compressed. So it's nice and it's rigid. Okay. Then it's also something that's necessary for anaphase and telophase. Okay. Because during this, what do? During anaphase, we're taking our sister chromatids, we're pulling them apart. So these are these fibers that are coming out and they're grabbing onto them. But when we look at this, we get an actin-myosin furrow here. That furrow is going to have the ability to actually split that cell into two identical daughter cells. So this is incredibly important for our cytokinesis as well. So that's our microfilaments. Let's talk about microtubules. microtubules. Microtubules are tubulin fibers. Okay, so when we're looking at these tubulin fibers, notice we have this interesting little design as we're looking at it. Okay, so when we're looking at this design, their whole thing is this is going to be the transportation highway, I guess is a way to think of it, transportation highway of the cell. And it's also within organelles. So it's going to be moving things up and down this highway, back and forth, so we can get things in the cell where they need to be. Microtubules are the basis of cilia and flagella. Okay? Both of these have a what we call 9+2 structure. Okay? You've seen this in your books. What the 9+2 structure is we have 9 pairs of microtubules with 2 microtubules here down in the center. That's our 9+2. Okay? Now, cilia is going to be moving things across the surface, whereas flagella are moving the cell itself. Okay, so moving things across the surface versus moving the cell itself. So we already talked a little bit about mitosis. Okay, in anaphase, we have those fibers that come out. They grab on to my condensed DNA, and they pull it. Well, that's microtubules. That's the main part of our mitotic spindles that come out from those centrioles, from those asters. They come out, and then they're able to contract and pull this back over so we can separate those sister chromatids. Okay, and look at the size of these. Very, very small. We're talking about 5 micrometers to 15 micrometers total size here. So very, very small. Now we talked about cilia, and cilia is going to be moving things within or around the cell. Okay, around the outside of the cell or within the cell. Now, cilia and flagella are different. We absolutely have cilia within our cells. We personally don't have flagella, okay, but we do have cilia, which means there's a clinical basis here for potential antibiotics, okay, because when we look at prokaryotes, they use protein flagellas, whereas eukaryotes, we use microtubules for our cilia. So the flagella is a promising target for antimicrobial drugs, okay, because it's specifically protein. It's not microtubules. It's slightly different. Sperm do have flagella. Sperm do have flagella. However, our flagella on the sperm are from a microtubule stance instead of from a protein stance, okay? So the thought is clinically, we should be able to leave the flagella on sperm alone because they're different, okay? Tom, it depends on what stage you're talking about within what we're looking at. So in anaphase, cytokinesis is where the microfibrils, okay, are going to be more active, microfilaments, okay, because they're actually doing that cleavage. During anaphase itself, where we're pulling on those mitotic spindles, that's going to be more microtubules, okay? So it's in anaphase that cleavage starts, but most of that cleavage actually occurs during our cytokinesis. So that's a really good question, okay? All right, so let's go ahead and look at intermediate filaments, okay? When we're talking about intermediate filaments here, we're talking about very filamentous proteins, and these are going to function in cell-to-cell adhesion, so the cell's ability to connect to another cell. It's also going to talk about our cytoskeleton integrity, okay? So when we look at this, we have polypeptides, okay? So we have this nice domain, we have tails, we have heads on them. Notice it's an amino head and a carboxy tail. Makes perfect sense, it's a polypeptide. Now, these can dimerize, and then 2 dimers can come together to form a tetramer. These tetramers will help us move into these filaments. So the protofilament they'll all start to come together to make these nice filaments. So let's talk about, when we say diverse, all of the different things that we can do within this. We've got our keratin. Keratin shows up in our hair, in our nails, in the outer layer of the skin. So it gives us this nice integrity. Okay, they're very, very strong. They can withstand a lot of tension. Okay, you can pull at your hair and it's not just going to break. Okay, so there's a lot of tension behind that keratin. That's that integrity. Okay, being able to do that. And then it also, with that integrity, can help us to anchor those membrane-bound organelles we were talking about earlier and the nucleus. So it can help us to anchor those things within the cell so that they're not going to be just freely floating around, freely moving around. When we're talking about these fibers, okay, these filaments, we're going to talk about something called fibrillin. Okay, fibrillin is a scaffold on which elastin is laid. If fibrillin has a mutation, we get a disease called Marfan syndrome. Okay, there is a rumor that Lincoln could very well have had Marfan syndrome because he was so tall and so long and lanky. So what happens when we don't have this scaffold that elastin is laid on is our connective tissue starts to not function correctly. Okay, that gives us those long spindly limbs that people think about when they think about Lincoln. Okay, excessively flexible joints, so nice beyond double joints. Okay, very, very flexible. It also can cause other problems like the lens of the eye can dislocate because that connective tissue is just not working the way that it should. It can also cause problems in heart valves and in the aorta because that connective tissue is not connecting things the way they should. So if we have a problem in some of these filaments, we can get diseases like Marfan syndrome. Okay, so we're really trying to bring in some clinical things tonight so that you can really see how just small things in individual cells or problems within individual cells can really cause larger problems within your patients. All right, let's go ahead, and this first question we're going to do together, and the reason that I want to do this together is I really want to make sure that we're using our question strategy of assess, plan, execute, and attack. Then for the second question, we're going to set you free on the second question on your own. Okay? So when we look at maternal inheritance, refers to genetic traits transmitted only from mother to child. Multiple metabolic diseases, especially certain devastating and neurodegenerative syndromes, show maternal inheritance. In these diseases, which of the following is most likely the primary cause? What is the one thing? So I'm thinking about this. It's got to be maternal. It's got to be something that I can only get from mom. So my plan is, what can I only get from mom? Oh, it's got to be mitochondrial DNA. Absolutely. So I know that that's the only thing I get from my mom. I can only get that from my mom. So the primary cause has got to have something to do with the mitochondria or the mitochondrial DNA, and that gives us C. I know, Lauren. Thanks, Mom. That's kind of a bummer. Okay. Mother's Day is coming up, so call and thank your mother for all the mitochondria that you have. All right. So for our second question, let's go ahead and, Eli, can you pull up a timer? And I think you guys can do this very, very quickly. So we're going to give you 30 seconds to do this, and then we'll pull up a poll. Good luck. Alrighty, guys. So what is that correct answer? Well, I'll tell you that about 89% of us have this correct. So autophagy is a process in which cells recycle materials. If I see recycled materials, I should be thinking about enzymes that can break those down. By degrading unnecessary or even harmful cellular components. So when we look at this, dysregulation of autophagy is implicated in cancer, okay, of certain diseases. Which of the following organelles most likely contributes to autophagy? So I'm seeing degrading, recycling materials. I should immediately be thinking, dun, 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 lysosome. Okay, so our lysosome here. All right, nice job with that. We are going to go ahead and look at just a couple parts of this question to just bring you focus on it, and then we'll let you go and do it for homework. So what we see is we see sterile regulatory element binding proteins. They give us the nice little shortened version of this. Our family of transcription factors that serve two functions ascribed to two domains. First, a sensor domain detects lipid levels inside of a cell. Okay. Second, in the case of low levels, it's cleaved to release a transcription factor that activates genes in lipid synthesis. Okay, so if I'm immediately, yeah, it's long. So I want you to focus on the fact that you've got two things in here that say lipid. And then you have the idea of transcription. Keep that in mind for when you do this question. Now, our next thing that we're going to do is look at our second learning goal. Our second learning goal here is really to understand how to classify prokaryotes. This is important because prior to things like PCR and ELISA assays, what we had to do is look at patient's blood or something taken from a patient, look at it under a microscope, and be able to identify it that way. We still spend quite a bit of time looking at things under microscopes when we are dealing with patients. Okay, so we really need to be able to classify our prokaryotes as well as look at the functions of prokaryotic cells. So we're going to start off by classifying by cell shape. So we've got cocci, we've got bacilli, and we have sporuli. So we're going to talk about those on the next page. This is important because we need to understand some of the differences between prokaryotes and eukaryotes. One of the huge differences is if I talk about ribosomes. Does anybody know the difference in ribosomes? What is a bacterial ribosome structure and what is a eukaryotic ribosomal structure? Yeah, it's absolutely the sizes. You guys need to know the sizes. So you need to know that if we're talking about prokaryotes, yes you do, when we're talking about prokaryotes, it's 30S and 50S. So the small subunit is 30S, the big subunit is 50S. Together it's called 70S. Okay? So 30 and 50 for prokaryotes, which means, what is it for eukaryotes? It's just 10 more for each of them. Instead of 30 and 50, it's going to be, yep, I'm making you do math, 40 and 60, which adds up to 80S. So our small subunit is the 40S, our big subunit is the 80S. Okay, so keep in mind the differences between those guys. Really make sure that you understand the differences in those ribosomes. So let's first talk about our classification. Three major types. First of all, we have cocci. Okay, so this is nice, it's round, it has a tendency to be in strings, like what we're seeing right here. And this has something like Streptococcus pyogenes, Streptococcus mutans. Streptococcus mutans is natural in our mouth. That's one of the things that actually makes dental caries, helps us to make our nice, wonderful cavities that we have. Absolutely. So Streptococcus, whether it's pyogenes or whether it's mutans, is a coccal gene. Okay, so we can see here that they can be in these nice strings. The next is bacillus. These are nice and these are rod-shaped. What is the most common bacillus that you guys should all know? I know you've all heard of it. There's staph bacillus. Usually we see staphylococcus as well. But yeah, the big one I'm looking for is Escherichia coli. So E. coli is the most common bacilli that we hear about always, always. Anthrax is also a bacilli. Hopefully you don't have to be thinking too much about anthrax in your career. Hopefully you're thinking more about E. coli and less about anthrax. Just saying, you know, positives and negatives here. Okay? So our bacillus here we can see is the pink, is these nice guys. And then we have sporulli. And sporulli are very, very interesting shaped. Okay? Treponema pallidum is a sporulli, and that's the sporulli that actually causes syphilis. Now, as we look down here, we see a purple and a pink. What is this staining? What is it called when we get this staining? Because we're going to be talking about it in just a second with the cell wall. Gram-positive and gram-negative. Absolutely, the gram-positive is going to really hold that crystal violet. That's going to be our streptococcus that we were looking at here. And our gram-negative is going to be our E. coli, our bacilli. Okay? So let's actually look at what we're looking for when we're talking about the structure. First of all, prokaryotes, they have no nucleus. Why? Because their DNA is in a nucleoid region. It's just a circular piece of DNA that we see in this nucleoid region. They have ribosomes, which we already said are 30 and 50S. Then they have a cell membrane and a cell wall. Okay? The cell wall is what gives us that gram-positive or gram-negative stain. Okay? So in the prokaryote, the genome, we just have that plasmid that is in a region. It's not separated out, and it doesn't need to be separated out. Then we also have with our cell wall, gram-positive cells are very, very thick. So they have peptidoglycans. Peptidoglycans, peptido means peptide. Glycan means sugar. Okay? So this is a nice, thick layer of amino acids and sugars. It really holds on to that crystal violet, and that's why you get that purple color. Whereas gram-negatives, they have a much thinner cell wall that are phospholipids and some liposaccharides. Because of that, we can wash it off and we get that nice light pink color. Okay, so that's what we're looking at when we're looking at our gram-positive and our gram-negative stains. Then we have our flagellum. Okay, we've got this nice area of motility. When we look at our flagellum, this long piece here is the flagellum, and it's hooked. It's got a hook piece that hooks it into this nice round guy. The round guy is called the basal body. And the basal body actually anchors this to the membrane and is the motor. There is a hook inside there that hooks that flagellum to that basal body so that it can rotate, okay, and it can have that movement that we need. This part, we should know what we're talking about here. We've already got this. You've already mentioned binary fission. This whole section is going to be about how do bacterial cells or prokaryotic cells reproduce, okay, and how quickly do they reproduce? How do they share their DNA? So binary fission is that asexual reproduction where they just split, the cells are identical. Transformation is the ability to bring foreign DNA or DNA that is outside that bacterium into that bacterium and be able to use that and utilize that. Conjugation is a sexual form of reproduction that uses a pilus. Transduction is viruses like a bacteriophage. And then our growth, there are 4 phases that we're going to look at as we go through. So first thing is binary fission. This is just a very, very simple form of asexual reproduction. What happens is the chromosome, that circular chromosome, is actually going to attach to the cell wall itself. So it's going to come down here, attach to the cell wall, and replicate while the cell continues to grow, continues to get larger. Then we get, once we have that replication, to the invagination. So the invagination is where that plasma membrane and cell wall are going to grow inward, and it grows inward along that midline. And we mentioned the midline earlier, and it's the same idea as when we have that nice midline when we're going through telophase, okay, and cytokinesis. At the end, we have 2 identical daughter cells, okay? Now, the pace at which binary fission occurs is different for each bacterium, and it can be very, very, very fast. Okay, Clostridium pharyngens is what causes food poisoning. Okay, Clostridium has a doubling time of 10 minutes. Okay, this is why food poisoning only takes a couple of hours to display itself, because it's got that 10 minutes for its doubling time. Let's talk about transformation. Transformation, we said, is being able to take foreign DNA, DNA that is outside the cell, bring it inside the cell and use it. Okay. The first place we saw this was Griffith's experiment, which you guys should all know about the smooth and rough bacteria. The smooth bacteria killed the mice. The rough bacteria did not. But if we had rough bacteria with heat-killed smooth, they pulled that DNA in, and they were able to become smooth. So it's something that bacteria do naturally. Okay? So that genetic material will go into the cell. We can also force them to do it not naturally in a lab. We can either do transformations with calcium chloride poking holes in the cell membrane and then heat shocking it, or we can do electroporation and electrocute them. Either way, what it does is it forces it to bring up that DNA. And that extra DNA will replicate by itself. Okay? And that allows us to put things into a bacterium that we want. Now, conjugation is looking at really asexual reproduction. And this is where we have F plus and F minus cells. So the F plasmid, the presence of this makes it an F plus. Okay, so an F plus is a male plasmid that has the ability to make a pilus. Okay, so this is going to then conjugate with this sex pili, and it's going to conjugate or hook together these two cells. Okay, so this is a unidirectional transfer. I can only transfer from here into the recipient. Cannot go the other way. If you do not contain the F plasmid, it cannot go the other way. Okay? Ah, they can. Absolutely, Brianna. So Brianna's asking, can they swap sexes? Absolutely. So it doesn't always happen, but the F plasmid can be reproduced, and it can pass that F plasmid into the new recipient. Doesn't have to. It can be other things that are transferred in there. And if that happens, that fertility factor, that F factor that makes it F plus, goes across that conjugation bridge, and then we end up with two that are F plus, or what we would consider the male plasma. Okay. An example of this is Shigella, which is dysentery, something that we don't ever want to have. What this does, what dysentery does, is it uses this to spread the R factor, and the R factor is the ability to be antibiotic resistant. So Shigella or dysentery, they can pass that back and forth between each other so that they all become antibiotic resistant. Transduction is specifically through a virus. Okay, the virus is called a bacteriophage. Bacteriophages only attack bacteria, so they're not going to harm us any, just like different viruses from humans don't attack dogs or birds, etc. Okay, so it has to be a very specific virus. The viral DNA is put into that host cell, and that's what these blue circles are. Okay, so diphtheria is something that does this. So the toxic gene in diphtheria is passed through a bacteriophage. Nice, huh? So we're getting all these wonderfully fun and exciting diseases here. When we look at transduction, so we have our bacteriophage. Our bacteriophage is going to put that bacterial genome into the cell, that viral DNA. So the blue is the bacterial, the pink is the viral. And what happens is when we replicate that bacterial DNA, because the viral DNA integrates itself, will also do the viral DNA. That happens in the lysogenic stage. And then in the lytic stage, once we start making all of this replication, so lysogenic, you don't see it, lytic stage, we lyse, we release, it goes to other cells and it can integrate DNA into the cells as well. So this is transduction. All right, well, now we know how they transfer genomes. How do they grow? Okay, and we said they have varying times. Okay, what causes food poisoning? Clostridium is 10 minutes. E. coli in perfect cultures, which means in the lab, is going to be a 30-minute. So when we look at this, here's our growth curve. So every time we see a graph, we look at the y and x axis. The y-axis is going to be the number of bacteria in logs. The x-axis is going to be time. First thing we have is a lag phase where the bacterium is getting used to the new environment. Okay? Once they do that, they start to grow like crazy. Whatever that doubling time is, they start to use all of the resources that we give them. At some point, however, once we get past this log phase, we're going to start to get that stationary phase. That stationary phase, they're no longer growing because in reality, they have used up their resources. Okay, so we're reducing those resources. If we continue to reduce those resources, we don't give them any more, they're going to go into a death phase, which is their final phase. Okay, so this is know your lag phase, getting used to the environment. Our exponential phase, woohoo, eating all that wonderful food, our resources. Stationary is where they stop growing because we reduce the resources, and then they will eventually hit a death phase where they will die. All right, so that's our depletion of our environment. All right, we have 2 questions for you to do. So here's what we're going to do. Eli, can you set a timer for 1.5 minutes? Okay, you guys should have this handout. Okay, so if you've downloaded it, go ahead and look at question 4 and 5. I will actually unsync this so you can move it forward if need be to get to question 5. All right, you've got 1.5 minutes. Eli's got our timer. Good luck. Alrighty, go ahead and put your answer in for number 4 if you have not already. I'm going to resync this. Molecular biologists commonly use plasmids containing antibiotic resistance genes to facilitate molecular cloning. These plasmids are not permitted to be released into the environment due to concern that bacteria will become antibiotic resistant. What mechanism do we do this? What do we do in a lab? We force it to take up that DNA. That is transformation. Transfection is putting a plasmid into a eukaryotic cell. So we didn't talk about transfection, but that is into a eukaryotic cell. Conjugation, remember, you have to have that F plasmid, that's sexual reproduction between them. And then transduction is looking at the bacteriophage. Question 5. Put your question number 5 in. So we have a student working with a wild type and 2 derivative strains. of E. coli. Normal growth of A was inhibited with a co-culture of B, which means B's got something or it's making something that A doesn't like. The culture medium of B also showed inhibitory effects after a 0.45 micrometer filtration, which only allows the passages of virons, which means if the virons are very, very concentrated, B has a problem as well. Growth of C was not inhibited by a co-culture with B or filtered medium of B. So C's got something in it that causes inhibition, okay? So it inhibits whatever that effect is, keeps it alive. Which of the following is false? Okay, we've got 4 things here. So 1 will be false, 3 will be true. Strain B carries the bacterial phage genome. That's true because it causes A to die. So that one's true. B, strain B could also express the restriction endonuclease C. If it has a restriction endonuclease, which this says C has, then B would not die with the filtered. So that is absolutely false. Okay, strain B releases phages that infect A. This is true. The phages released by B could infect strain C. This is true. Could infect it, but it doesn't matter because it has that ability to have the endonucleases. So that one is B. All right, finally, what did we do tonight? We had a couple of different things we went through. We looked at our membrane-bound organelles and our cytoskeletal structures. Then we looked at our different bacteria, different shapes of bacteria. We looked at the cell wall and flagella of bacteria, and we described binary fission, transformation, conjugation, and transduction, and how our bacteria grow. All right, thank you so much for joining us. Hopefully we'll see you in the kidney section. Have a great night. Bye.